In the last lecture, we learned about the basics of motor control, primarily focusing on the three basic movements: reflex, rhythmic, and voluntary movements. But a lot of that was focused on how the spinal cord contributes to that process and how the upper motor neuron system, primarily regulated through the motor cortex, allows for voluntary muscle control, with a lot of the focus on the limbs, arms and legs. Today we're going to be talking about a very specialized motor control circuit, and this is how the muscles control movements of the eyes. When you talk about any muscle system, you can break it down into further and further components. And when you talk about the musculature of the face, the head, and the neck, things get really, really complicated. And that's because our faces and our heads do have an awful lot of fine motor control that we don't necessarily think about. Obviously, our heads can turn in a variety of directions: up, down, left, and right. There's complex muscle systems that control that. We also have a ton of tiny little muscles within our face to control facial expressions, chewing, talking. Some of us can even move our ears. We have various glands that also have some basic smooth muscle control. All of this has to be coordinated by the nervous system. So therefore, we have a very complex network of facial nerves that innervate these muscles and help coordinate them. I don't expect you to have any of this memorized. I'm showing these diagrams just so that you can appreciate the complexity of this overall circuitry. We have dozens of muscle groups within our face. We have a variety of nerves and nerve branches that have to innervate and control each of those muscle groups. This is a simplified version of the face. To show you just how complicated it can get, here's a different diagram just showing innervation of the lower part of the face and in the neck. You can just see how complicated this can get. Today, we're going to be just focusing on a very small subset of overall muscle control involving the head, and that's going to be eye movement. I wanted to provide another revisit to the facial nerves because these are critical for understanding how the nervous system is organized and controls the major groups of what's going on within the head. We spent a lot of time talking about the sensory inputs. We have talked about the optic input, the cranial nerve 2 with the optic nerve. We have also discussed those that are involved with taste and smell. We also talked about hearing, cranial nerve 8, which is the auditory vestibular nerve. Those are the major sensory inputs that we have discussed, and that will be it as far as what we discuss for those. There are now other cranial facial nerves that control muscles, and there are three that we're going to be talking about today. That is the oculomotor cranial nerve, the trochlear cranial nerve, and the abducens cranial nerve. These are cranial nerves 3, 4, and 8. We will talk about those in a little bit more detail in a few more slides. First, we got to talk about eye movement. So this is a visual field, nothing on it. Your eyes are probably just kind of wandering around, wondering why I'm showing you a blank space. Ah, there's a heart. There's another heart. So I'm throwing things up in this visual space. And your eyes are probably now going between the hearts. When the first heart picked up, your eyes attended to that. Then the second eye attended to that. Now that heart on the left moved. What are your eyes doing? Have you ever thought about that? Have you ever thought about what actually happens when things just suddenly pop up in visual space, or when things are moving within visual space? This is a pretty mundane question, but how does the nervous system allow for us to do those things? And it turns out there are specific circuits that control these things. And we're going to learn about those today. Our eyes have two basic forms of movement that we can put all eye movement into two basic categories. First are saccades. These are fast ballistic movements to a target. These are super fast. They can be finished, over and done with, less than 50 milliseconds, even shorter than that. Then we have smooth pursuit. So this is what we were using when that heart was moving across the screen. This is a more deliberate, gradual movement to track objects in space in order to stabilize the image onto the retina. So let's first talk about saccades. Generally, this is how we observe visual space. Our eyes dart from feature to feature when we are looking at even a picture or a face, especially in faces. There's a certain pattern that neurotypical individuals will use to observe faces, going from eye to eye. What our eyes are doing is that we will focus on a feature, and then our eye will dart suddenly to another feature and focus on that a little bit, and then dart down to another feature and focus on that, dart to over here to the mouth, focus on that. This diagram is what was used. This is showing what was one particular human was wearing an eye tracking system, and the length of time is illustrated by how dark the dot is, like how big that dot is. And you can see essentially what we have here are very small thin traces with going to specific dots, creating a tracing pattern that goes around this face. And you can see this person, and this is true for most individuals, tend to focus on the eyes, going back and forth, back and forth, back and forth. Our brains, we don't think about this quick dotting eye movement. We're constantly doing it all the time, and this is how we assemble our cognitive perception of a visual field, of seeing a face. We don't necessarily think about that we're focusing on one eye or the other eye at any one time. Instead, our brain is just constantly moving around, and so that we can assemble what that looks like in our brains at any so that we have a cognitive understanding of what we're actually looking at. So that's true for this kind of face. This is true for faces in profile. You can see that, again, people tend to attend to facial features, even in profile. But we also just look at contrast, interestingly, right? So our eyes are really designed to pick up edges, and we can pick up edges very, very well. And you can see that the eye is tracking individual points of contrast around an image in order to assemble it so that we have a cognitive reassembly within our cortex of what that actually looks like. Allows us to memorize these things and to understand these things. So saccades allow us to not only assemble what a visual field looks like, it also allows us to identify new targets in visual space. We make saccades when a target suddenly appears within the visual field. So this person, his head is pointed forwards, but his eyes are slightly turned to the left, and he's staring at this blue dot, calling it the old target. Then all of a sudden, a new target appears in his visual field. His eyes will suddenly quickly move over to that new target. We can measure this. There are a few data points that you can get from this. One, we can look at overall eye position, right? So his position, so we're going to say that in this case, there's about 10 degrees difference in the visual field between old target and new target. And his eye position was at the 5-degree mark when it was looking at the old target towards the left. And then all of a sudden, when the new target appeared, there was a little bit of delay, which we'll talk about in a moment. And then his eyes moved over to the right. So eye position went from left to right, around 10 degrees, according to this data plot. We can also measure the velocity. How quickly are his eyes moving, right? That's essentially just change in position over time. And you can see that there's a ramp up and then pretty steady velocity. It's super quick, and then all of a sudden it has to decelerate and maintain its position. And so that's a change in velocity. Velocity is peak at this moment here, right in the middle, and it's very short. You can see 100 milliseconds is this length from this point to this point, and this is about 20 milliseconds. So it is moving over a period of about 20 milliseconds, and then it can target precisely this new point. Our eyes are constantly doing this when we examine any visual field, and we have a tendency to move our eyes to focus on objects that are moving, especially objects with high contrast. Okay, so we're going to make saccades here. This is going to be fun. So we've got a heart there. We're going to do exactly what we just saw. We're attending to that heart. Take a look at the heart. All of a sudden, there's a new heart. Our eyes moved. This is exactly what we had just seen in before. And your eyes, when it popped up, when that new heart popped up on the right, there was a brief pause. And then it took about 10 to 20, maybe 30 milliseconds for your eyes to move from the one heart to the other heart. Very exciting. You made a saccade. Congratulations. So we're going to talk about how the visual system, specifically the visual motor control system, allows for doing that specific behavior. So there are a few ways that we can illustrate this. And now we're going to talk about the perception and tie into movement and the delay that has to occur in that process. So your eyes move from the left to the right. This is eye position. We already just described this. Your eyes were looking at the left, and then a new target popped up. Here's when the new target suddenly appeared. It's computer, so it's instantaneous. There's no delay whatsoever. Pops up. There's about a 200 millisecond delay when a target appears for the eye to suddenly react. And then when it reacts, it takes, like I said, around 20 to 30 milliseconds to actually move to the new target. And then it'll be precisely aligned with that new target. So this is a behavior described by Albert Fuchs many years ago. We knew about saccades before that, but he did an awful lot of the understanding of the neural circuitry that controls this. I only mention him because he was actually the director of my PhD program when I was getting my PhD at the University of Washington in Seattle. And he was a wonderful person, a wonderful director of our program, and has done an enormous amount of work on this process. Everything that we talked to up until this point, for the most part, has been about saccades, and that is what we're going to mostly focus on. There are other forms of visual control of eye movement that we put into the category of smooth pursuit, and this involves gradually moving the eye position to track a moving target. So in this case, this red dot is going across the visual field, and our eyes can maintain that tracking. And you can see this with the eye position. Instead of a sudden movement and then maintaining to a new target that's static in space, not moving, instead our eyes are tracking the target. The target may start to suddenly move, and there'll be kind of a quick catch up with the eye velocity, so it has to go a little fast to catch up. And you can see it moves a little quickly, and then it just maintains the straight line because this object is moving at a constant velocity. This is the basic form of smooth pursuit. We can put smooth pursuit, what our eyes do when they're moving gradually, in two different categories. One is called the vestibulo-ocular reflex. So this involves with coordination of the head and the eyes. Vestibulo, because it's referring to change in position of the body, in this case the head. So this is what happens when you're staring forward at a particular point, and you can do this right now, looking at the screen. You can look right at this line, you're looking at it, and now just kind of gently move your head to the left. Move your head to the left, but keep looking at the screen. What are your eyes doing? Your eyes are still maintaining that spot in the visual field. You can see this little red dot. Look at that. Move your head. Your head is rotating this way, but your eyes actually have to rotate in the opposite direction. Your right eye is going towards the temporal side of your head. Your left eye is going towards the nasal side of your head. You're maintaining the focus on that, but your eye position actually has to move in the opposite direction. So head rotation goes in one direction, and your eyes do counter rotation in order to do this. And this is a coordination of the vestibulo-ocular reflex system. There's another form of smooth pursuit, which is the optokinetic response. And so this is when things are moving in visual space. And this is another thing that we just naturally do. If a train is going by, or if you're driving and or as a passenger and you just kind of look out the window and things are just going by in visual space, you will be doing a repeated optokinetic response where the surroundings are traveling and moving and your line of sight is following the surroundings. So let's say you're in a parked car, all of a sudden the car starts moving, your eyes will have a 50 to 100 millisecond delay, and then all of a sudden it'll start moving, and it'll be moving at the same speed that the surroundings are moving. This is essentially the same thing that's happening with smooth pursuit when you're tracking a moving target. That's the optokinetic response. We're not going to talk about any of this in great detail. I will briefly talk about the vestibular-ocular reflex at the very end of this lecture, because it's an interesting circuit, and I'm not going to walk through it in super great detail. It's not going to be on the exam. It'll be extra material, so you could stop watching right at that moment if you don't want to hear about the vestibulo-ocular reflex. So let's do some smooth pursuit and talk about a little bit more some additional features of smooth pursuit. All right, so we've got a heart on the right. Let's bring this over here. Look at the heart and stare at the heart. Okay. It popped up, and then you started tracking the heart. There are something called catch-up saccades. So that sudden movement over to the left and then doing smooth pursuit, that is a saccade that you actually made because all of a sudden you had a new target that appeared in visual space. Catch-up saccades allow for precise tracking. So we can change the speed of our new object and see and monitor how the eye position over time and see how well it tracks the object. It turns out it's actually incredibly accurate, but there's a little bit of a delay that occurs. So what we're looking at here are these red lines of these different objects, and they're traveling at different speeds. One is traveling at 10 degrees per second, another one is traveling at 15 degrees per second, and the other one is traveling, the third one is traveling at 20 degrees per second. So here's a second right here, right? 10 degrees. It traveled 10 degrees. There's 10 in 1 second. 15 and 20. And that's just a speed, right? So at half a second, this is at 5 degrees, 7.5, and 10, right? Because it's at a constant rate. When these things suddenly appear, they start moving at their rate. Our eyes have a little bit of delay in order to see that. So this is tracking eye position, and you can see that there's a very quick saccade to where the object is now in space, and it actually moves very precisely to where that object is now located. You can see that the eye had to move to here at like around 2.6, and here it was about 3.4, and then here it's about 5.8. It was able to target precisely where these things were moving and then track it precisely, almost instantaneously, tracking the object without any sort of hiccup or delay. And then we can track at different speeds. The eye is moving along with the object. It's going either slow, medium, or fast. But there's this catch-up saccade that has to occur there. So again, this is more work by Albert Fuchs, described it in that same paper from before. Yeah, really interesting way that catch-up saccades can allow for very precise tracking of objects in space. Up until this point, we have talked about a variety of eye movements, so of course it's going to be really important for us to learn about the muscles that control the eyes. There are 3 pairs of muscles for each eyeball. We have in the horizontal direction the lateral rectus and the medial rectus. Lateral will be towards the temporal side, medial towards the nasal. And then we have muscles that control our eye position in the vertical direction, which is the superior rectus and the inferior rectus. We also have another pair. The third pair is the superior oblique and inferior oblique, which is really important for binocular vision. We are not going to talk about those at all. Instead, we're going to focus on these simpler muscles. You can see it's a nice coordinated horizontal or vertical, the lateral rectus and medial rectus, superior rectus and inferior rectus. Superior means up, inferior means below, lateral is toward the side, medial towards the midline. Pretty easy, and then all of them are rectus. Now, one thing that's to make it a little bit more complicated is that the eyes are not identical. So this is this person's right eye, and I realize, like, okay, now you're looking at someone's eye and the muscles are been pulled out and it's pretty freaky looking, like something out of a horror movie, but it's a nice way of illustrating the motor control. So this right eye, the lateral rectus is towards this person's right, but if you're looking at the left eye, it's the medial rectus that is towards the right of the left eye. They are mirrors of each other, just like how the retina are mirrors. This is also true for the eyes. So think through this a little bit. When say this person is moving his eyes to the right, that means that the muscles in the eyes have to be doing opposite things. It means that the lateral rectus will need to contract and pull the eye to the right. It also means, and that's true for the right eye, it also means at the same exact time that the medial rectus has to start firing on the left eye and pull the eye to the right as well. So this eye has to go towards the nose, this eye has to go towards the temple. And then if you go eyes to the left, you have to have the opposite happening. So this is really complicated. And we will be talking about the neural circuit that controls this. There are 3 cranial nerves that we have to focus on for this. One is, the first is the third cranial nerve, known as the oculomotor nerve. That is located here, cranial nerve 3. There's also cranial nerve 4, which is the trochlear nerve. And then there's cranial nerve 6, the abducens nerve. That's this one here. The muscles of the eyes are innervated by these 3 nerves. The oculomotor nerve, it gets its name because it innervates the most muscles in the eye. It innervates the inferior rectus, the medial rectus, and the superior rectus. The abducens nerve innervates the lateral rectus. And then the trochlear nucleus innervates the superior oblique. So we will not really be focusing on that at all. We're going to be talking about mostly activity from cranial nerve 3, the oculomotor nerve, and the abducens nerve. We are going to focus also, because we're not talking about how the eyes control binocular vision. We're going to ignore that completely. We also are not going to talk about moving the eyes in the vertical position. Instead, we're going to be focusing on how eyes move left and right. So just in the horizontal plane. We're going to talk about now how the pairs of muscles that have to contract, how they contract in order to move the eyes to the right for this individual. So again, remember, this person, if this person's moving his eyes to the right, the lateral rectus in the right eye needs to be contracted, and the medial rectus in the left eye needs to be contracted, because then both eyes can then rotate in the rightward direction. So we're going to walk through these steps. The first step is that there's input, which we will get to, that causes the right parameridian pontine reticular formation, or PPRF. It causes those neurons to fire. So there's input that's causing this nucleus to start firing, the right PPRF, okay? And we have to pay attention to whether we're talking about the left or the right. That is really critical to this. So, and to orient ourselves, just to back up a bit, these are the cross sections within the midbrain, where you would find the ocular motor nucleus, and so that would be right here. And then this is in the pons, which is below the cerebellum. And so we're looking at a cross section here,
and this is the midline. So this is the right PPRF, and those neurons are firing. Those neurons project to the right abducens nucleus. So these neurons are firing, they're excitatory, they cause neurons in the right abducens nucleus to fire as well. So let's pay attention to this green neuron first. This neuron is firing. This makes up the abducens nerve, which is synapsed onto the lateral rectus of the right eye. So this is starting to contract. There are other neurons within the abducens nucleus that project to the left oculomotor nucleus. So this decussates and then synapses onto the left oculomotor nucleus. Okay, so we're going from the right to the left. That left oculomotor nucleus synapses onto the medial rectus in the left eye. So now this is contracting. Now both muscles are contracting, and then the eyes are being pulled to the right. So you have this initial starting point, and then you have a coordinated system of saying, okay, well, we need opposite muscles to respond here. So this number 4 here is a critical component to this, where it takes the signal saying, hey, move to the right. If the right PPRF is firing, we need to move to the right. But we also got to get this eye moving towards the nose. And so you have another neuron that causes the left oculomotor nucleus to fire, causing that to then move to the right. So now the left eye is moving to rightward, and the right eye is moving rightward. And it's this simple, elegant, coordinated system that allows for that. That's the contraction part. We also have to talk about how these muscles relax, which we'll talk about in a couple of slides. So let's talk about firing of the abducens nerve and how this is correlated to how much movement is actually occurring and overall position of the eye. So we're talking about just this nerve here, the abducens nerve and the abducens neuron. So we can record from this. You can find this nerve and you can measure how many action potentials this nerve is firing. So that's what we're doing here. We're recording a neuron in the abducens nucleus. We could place an electrode there in the abducens nucleus. We know that this nerve is connected to the lateral rectus muscle. So this is what the muscle looks like when it's not disconnected and splayed out like we've seen in the previous diagrams. It would be here. And you can see if it contracts, it's going to pull this eye this way. So let's see what happens with the abducens nerve when it fires. So when the eye is just sort of in a center position, it's like this. And it's not really firing. This is our starting point. Now this eye is given a target. It's got to move, and it needs to move to a new position. The abducens nucleus fires, causing this muscle to contract, change its position, and then it kind of stops. So there's no firing happening here. Now we have another target, causing the eye to move lateral even more. So we need more firing to cause the eye to move. And now something interesting is happening. We're at a position that's kind of where it's not really like relaxed and comfortable to maintain that eye position. So now the abducens nucleus needs to continue to fire at a relatively low spontaneous rate in order to maintain this position. It's got to be pumping neurotransmitter in here, acetylcholine, to make sure that this muscle is contracting at a certain rate to maintain this eye position. Now we give it another target. It's got to move even more towards the lateral side of the head. And now the spontaneous firing rate is even higher than it was at this position. So because the eye is now moved very laterally, in order to maintain the position, the abducens nucleus also needs to fire. Now we're given it a target where it can now relax a little bit and go towards the medial side. The abducens nucleus quiets up. It stops to allow for the medial rectus to contract, right? So this has to quiet up. And then you have, we're at this new position, and you can see that this is in between the firing rate of these two. So this is relatively low, this is relatively high, and this is somewhere in between. This is the Goldilocks. Okay. So the firing of the abducens neuron, it's intuitive, but it actually has different states that it needs to maintain, whether it is moving laterally, it's got to fire a lot. If it's moving medially, it has to quiet up completely. And if it's maintaining a position at the lateral side of the head, it has to have a spontaneous firing rate in order to maintain that position. All right, so now we need to make this a little bit more complex. Hopefully you now fully understand what's going on here. If you don't, maybe you should go back a couple of slides and look at it again, because now we're going to make it even harder. So we talked about movement to the right and the lateral rectus and the medial rectus in the right eye and left eye respectively, and how they have to work. We need to relax the medial rectus in the right eye and the lateral rectus in the left eye. How does that work? So we're going to walk through that process. The right PPRF, this is where things really start for this circuit. Okay, so the right PPRF, we already talked about how that fires, right, in order to get everything going to the right side. So if the right PPRF is firing, we're going to move to the right. But it also helps coordinate the relaxation of the other muscles. It also synapses onto, and it's not within this diagram here, so I just added a neuron here to what's called the medullary reticular formation. It synapses onto this formation, and in the medullary reticular formation, there are inhibitory neurons. So it's going to cause inhibitory neurons in the medullary reticular formation to fire. The medullary reticular formation synapses onto the left abducens nucleus. So it has inhibitory neurons that come over here and synapse onto the left abducens nucleus. So now the medullary reticular formation is going to decrease the firing rate in this nucleus. That means this neuron that innervates the lateral rectus in the left eye relaxes. It's being inhibited by the medullary reticular formation. And remember, there are neurons within the left abducens nucleus that cross to the right and connect to the right ocular motor nucleus. And so then this neuron also decreases its firing rate, which then means that this neuron that's being fed by the left abducens nucleus, now on the right ocular motor nucleus decreases its firing rate, and then this muscle relaxes. So it's with this coordination system that allows for, you can see, we've got all the neurons here. We've got the right PPRF as the starting point. It causes these muscles to contract. And then because it also then brings in an inhibitory switch where you can flip the signal, which is done in the medullary reticular formation, it then causes the other side of the system. This is just a mirror, right? You can see it's essentially the same neurons. It's just we have to pay attention to whether we're talking about the right or the left. So in this case, the left is being inhibited, but then we also have to cross back to the right in order to get the left, the medial rectus in the right eye to relax. Walk through it a few times. It's actually not that complicated. You just have to think about, oh, well, now we have an inhibitory switch that's going to cause the opposite side to relax. So this is how that's all coordinated. Now we're going to talk about how the targets that appear in visual space cause a saccade. So we've got the basic circuitry of what is happening within the brainstem and the cranial facial nerves that control the muscles. And then we've also talked about how the PPRF is key to that, allowing for eyes to move either right or left and coordinating the muscles of the eyes, which are mirrors of each other, to allow them to move to their position, which is going to be opposite relative to where they are in the head, right? One eye moving towards the temple, the other eye has to move to the nasal side. This is because, so the eyes can do this is because we have retinotopy throughout the visual system, which we learned about in our visual system lectures. So this is taken from there, the visual system lectures, where we were talking about if you record within the visual cortex, you will find in a cat, if you're looking at a cat and you're recording in the visual cortex, there'll be visual space. That's what the screen is. And this is the center of this cat's visual space. You will find a place within visual space where if you place a bar at a particular orientation, it'll cause that neuron to fire, right? And then if you change the orientation, the neuron won't fire. But if you were to place this bar down here, it's not going to fire, even if it's at the right orientation, it's in the wrong place because neurons in the visual cortex are organized with a retinotopic manner. So we got to, not only do we have to pay attention to whether information is coming from the left or the right, we need to know where it is within the visual field. Is it towards the center, towards the fovea, or is it way out here in the periphery? And all of that information is maintained through the visual system. It's true, of course, for the retina because that's where the image is coordinated, and all of that information is maintained through the lateral geniculate nucleus, as we learned about, with the different having that volcano shape with the fovea being towards the center, and then the lateral geniculate nucleus maintains that within the visual cortex. Now, I didn't include this figure when we talked about the visual system, but I just want to reiterate what this looks like when within the primary visual cortex. So this is the visual field here, and we have our center gaze, which is controlled by the fovea. The fovea is where we have the sharpest images, and that's because where we have the most cones, and we have the least convergence of photoreceptors to bipolar cells there. Whereas in the periphery, we have a lot of convergence, but we can still see what's happening in the visual field there. So in the fovea, if you were looking, say, in this part of the visual field on the left-hand side, and you're looking at the upper left portion of our center of our gaze, you can track that information all the way. So over here, this would be this side, seeing the left part of the visual field. You can track it all the way to the right cortex, and there'll be a part of the cortex where you can record from neurons that will be sensitive to objects in number 1, bars especially. So if you put a bar with the right orientation, this is the part of the cortex that will see that. Let's say we put a bar in 9 in this part of the visual field. You'll have to look in a different part of the cortex. You'll have to look over here. And then, of course, the same is true when you look at the right visual field. You have to look at the left cortex, and you'll see a pretty similar pattern with the fovea towards one side and the periphery towards the other side. So there is this map within the visual field, retinotopy maintained within the visual cortex, but there is also retinotopy maintained in other important targets of the eye, which is the superior colliculus. So this is where we're going to talk about the superior colliculus. This is also organized with a retinotopic map. However, the superior colliculus gets direct input from the retina. So the retina actually synapses directly onto the superior colliculus. So let's talk a little about where the superior colliculus is located. So this is a medial cut of the brain. A lot of different features on here, a lot that we have talked about. Here's the thalamus right here. Here's the midbrain. Brainstem is over here. Here's the cerebellum. This is called the corpus callosum, which connects the two cortical hemispheres. We'll talk about that and how important that is for coordinating information across the brain when we talk about language. There's a bunch of other things here. Yeah, the optic chiasm we talked about. This is what it this is its position in the medial part. And we'll be revisiting this when we talk about the hypothalamus in a couple of weeks. There's also something called the inferior colliculus, which I mentioned when we talked about the auditory system. The superior colliculus is critical for the visual system, and it helps to control eye movement as well as head and neck muscles. And we're going to talk about its role in controlling eye movement for really most of the rest of today's presentation. So here are a few images describing the organization of the superior colliculus. So this is oftentimes you will see in papers, and this is in a research paper that I took this figure from. Here's the aqueduct. So this is part of the ventricular system. And this is the midline. And in fact, PAG, what is this? This is the periaqueductal gray, which we learned about for the descending pain pathways. Then, right near the periaqueductal gray, we can also find the superior colliculus. And so that's what we're going to talk about. So that is this portion here. It is organized in layers. There are layers through the superior colliculus. And the thing about the superior colliculus is that it is a coordination system. Like I said, it receives input from the retina, and it also has a retinotopic map. So there are different layers, and you don't need to know the layers. You don't need to have them memorized, like what the names of the layers are. There are specific names. We don't need to know them. But what you do need to know is that the retina, here we have the temporal side of the retina, the nasal side of the retina, and we see this snake crawling in front of us, freaking us out, which is one important reason why we would need a reaction system like this. We perceive the snake. It hits the neurons within the retina. This information is, of course, sent up through the lateral trabeculae nucleus to the V1, but it also is sent directly to the superior colliculus and synapses onto layers within the superior colliculus to have a very specific map where there will be a temporal side and a nasal side. You don't need to know which side is which. You just need to know that there's a retinotopic map within the superior colliculus, that there are neurons that will synapse. So like here's this temporal neuron, it is synapsing on this part here, and then the nasal part is synapsing way over here. There's a retinotopic map, and not only do we have within a nasal temporal portion, we also have vertical direction as well, with the superior and inferior parts of our retina being transposed as well in the different dimension. So there's a retinotopic map. We also receive input from V1. So V1 is also coordinating this, and it's maintaining its retinotopy. So you can see this part of the visual field is sensing this, and this part of the visual field is sensing this, and it's being maintained at a different layer. So we've got coordination of visual information coming into the superior colliculus. But that's not it. We also have information coming from the somatosensory cortex that goes in here. And so that there's a somatotopic map within the superior colliculus. We also have auditory information that comes into the superior colliculus, giving us that information that's really important for where sounds are coming in space, is also coordinated in here. It's not illustrated in this figure, but we will learn about that when we talk about the superior colliculus in barn owls in a little bit. So there's a map. There are a bunch of maps, and the superior colliculus is a critical portion of our nervous system that allows for comparison of sensory information from multiple modalities: somatosensory, visual, auditory. The responses of the visual field are coordinated with information from the auditory inputs as well. So you don't need to know the orientation here of A, B, C, and D, but what I want you to see is that there is a map here. So we've got the visual system with A and B located here. So this is our snake with the, this is the nasal portion. This is the temporal portion. So B is located here in this part of the superior colliculus. A is over here. And of course, we also have the vertical dimension, which is gets less representation. We are, like most mammals, including humans, we tend to attend to things happening more horizontally than vertically, and our nervous system reflects that. Big surprise. But we also have an auditory map, right? That part of the MSO that helps the coordination of knowing where things are located in space. That is mapped also within the superior colliculus, and it is also coordinated in that same way. So just illustrating this a little bit further, that the auditory map is coordinated with the visual map, which also gets input and is especially for animals that use somatosensation, like animals that have whiskers. There's also a map there that includes that information too. Okay, so that's about the superior colliculus and its overall basic organization. So what's up with all these layers? You've got the visual information, there's auditory, somatosensory. When you get deeper into the superior colliculus, now you start to get into a different set of neurons that are motor neurons, or they are involved in motor control. So the superior colliculus has this map, and it will see things pop up in visual space, for instance. And then that visual information, this is where that visual information appearing in space allows for coordinating a motor output. And it happens through these different layers. So you got the visual layers at the top and visual input coming into that space. And here we're showing what's happening in here. So we've got the target position. All of a sudden something appears. And if we are recording from the visual layer, so we've got an electrode that's allowing us to record from a visual a neuron in the superior colliculus that's within that part of the visual field. We're recording that neuron. And then we also are recording from a different motor neuron, and we know that these things are electrically connected. So and we're recording this from a live animal, so we're recording from these two different layers. When you present a target, so all of a sudden a target appears like an orange heart, what we see in the visual side is that neurons in the superior colliculus start to fire. It takes a little bit of time for that information to get to the superior colliculus, but they start to fire. And then, all of a sudden, before the saccade occurs, the motor layer will start to fire as well. So the visual information is being passed from the visual layers and coordinating the motor layers to start firing. And this is where that transfer of sensory information to coordinate motor output is occurring. And it's allowing for saying, hey, something appeared at this place in the visual field. You need to move this much, your eyes this much in order to see it. And that allows for this coordination of visual information within the map to get precise movement of the motor output. We can induce these things too. So that's one way of just recording. In here, what we're doing is we're looking at stimulating these visual layers and then seeing what happens in the output. So we're recording here. That's what we're seeing here. We can measure current and voltage. And when we stimulate, this is the stimulation artifact. So if you just do like a single pulse of this visual system neuron, you'll get inward current within the neuron that we're recording from in the motor layer. And we get EPSCs, excitatory postsynaptic currents. We can also see action potentials that occur when we stimulate. So you can show that these neurons are electrically coupled. And then not only that, when you are doing these kinds of recordings, you can put in biosightin, and some of this is described in the previous slides. You'll have to go back and look at the description that's in there. I included the entire caption just so that you could read through it. But we can fill these cells. And so these are actually like drawings of actual neurons that were recorded. And we can see that not only are they electrically coupled, but when we go back and we fill them and then we cut, when the animal is killed, we slice the brain, we can stain to see what the shapes of these cells are and then trace them out. And that's exactly what we're looking at here. And we can see that they're actually connected as well. So these neurons are synaptically coupled. That's what this is a demonstration of, that if you stimulate here, you can induce firing here. And then not only that, if you're recording here, because you can stimulate and record with the same electrode, and you present a stimulus, you see activity here, followed by activity here. And the way this works, of course, is that we've got the retina coming in. This is now the transfer of the visual information to motor output, which then goes to the gaze centers to then induce a saccade. So the output motor neurons in the superior colliculus, they synapse onto the neurons in the PPRF. I don't expect you to know this circuit in any detail. So these are the different layers of superior colliculus and how they're connected to, believe it or not, the PPRF also has different parts and is somewhat complicated. But this is the whole PPRF. This is the superior colliculus, SC here. And this is a diagram showing how that there is this coordination of activity from the superior colliculus directly synapsing onto the PPRF, which we learned about is the key starting point of controlling the muscles. And so the superior colliculus has the visual map that then tells the PPRF how much the eyes need to move in order to move the eyes in a precise place. So if you feel like it, you can kind of go back and work your way through with this. This is the simplified version of this, where you have neurons in the superior colliculus connected to the PPRF that allows the control for that. So a little bit more about this map, because I think that it's really important that you come away with this understanding of that we have a visual field, but within the visual field that allows for the coordination of movement of the eyes, okay? We can record, so this is a simplified diagram of recordings that had been made within the visual portions of the superior colliculus. And remember, we have superior colliculi on both sides of our brain. We have it on the left-hand side and we have it on the right-hand side. And this is kind of a nice illustration of how this is mapped within the superior colliculus. Okay, so in here, this is the superior colliculus, just like most of the visual field maps, they're somewhat distorted, with the center of our visual field getting the most representation, and then the more peripheral parts getting less representation, and they make up a smaller part of the visual field. And the receptive fields are smaller the closer you are to the center of our visual field, and they get bigger the farther away you are. The superior colliculus is also organized with a split, with the left-hand side of our visual field being coordinated in the right, and the right side of our visual field being coordinated in the left. So these neurons are decussating at the optic chiasm to synapse on the contralateral superior colliculus. The black line here represents the horizontal line here. The blue represents the midline in vertical space. And then, so this is the midline, and then we're moving away from the center out towards the outside. And you can see, like, 5 degrees is here. That is right here. This is 5 degrees, and you can see it takes a pretty big chunk. And then from 5 to 20, so we go from 5 to 20, that's here. So this 20-degree line is this. And then from 20 to 40 is out here. That's true for the left, and it's mirrored on the right. So 0 degrees is right here. This would be the origin, with the blue line being this. And then red is the right side of the visual field represented in the left superior colliculus. We can record from neurons within the superior colliculus and see what's happening. And we can also induce activity in these areas and see what happens to the eyes. And so that's what this figure is illustrating. So let's take number 1, for instance. So we're recording from neurons there, okay? And we are trying to figure out where in visual space is this receptive field. And we map it out, and it turns out the receptive field for this is relatively small. This is number 1, and it's pretty close to the center of our visual field. And you can see that that's true here, right? It's relatively close to the part where that would be that part of the retina. So that's the map. The other thing that you can do with this is that you can tell the animal to maintain his gaze at the center of his visual field and then stimulate number 1. So the animal is being told, hey, you got to maintain your gaze here. If you tell the animal to maintain his gaze here, but then you stimulate at number 1, you can cause the eyes to move there. So not only are the neurons sensitive to objects appearing here, so if you put an object here, the neurons will fire, but if you stimulate, you can cause the eyes to move, which is showing that the coordination of the retinotopic map is inducing precise movements. And this is true for any of these parts. So let's go through number 2. This neuron has this receptive field, so any object appearing here will cause these neurons to fire. But if you tell the animal, okay, maintain your gaze here, and you stimulate it, the animal will automatically move his eyes to that part of the receptive field. The same is for 3, same as for 4, and then you can do it to the other side. So if you record from the other superior colliculus in the right-hand side of the brain, and then you start looking in the left visual field, you can find neurons that have these receptive fields, but if you stimulate, you can also cause them to move to those receptive fields. So this experiment really nicely illustrates that not only do we have this map of the receptive fields within the superior colliculus, if you stimulate those neurons within that map, you can cause the eyes to move to those precise locations. So this is where that all occurs. And it's the superior colliculus that coordinates that movement. This coordination also involves the auditory system. I am not going to walk through this in great detail. There's been a ton of work on how the auditory space coordinates with motor output as well as overall visual space. And owls have very good eyes, but they also have very good ears, and they coordinate these things in order to do hunting, because they hunt at night and they can identify where animals, prey items are located through hearing. And then the precise coordination of catching those prey items, like a mouse, also has to involve the visual coordination as well. They have very good eyes and very good ears. And there is a map of the retinotactile input onto the auditory input as well. And that this map in auditory space coordinates with the visual space. And some of the work that's been done with the barn owls, and I only mention this because there's been some work to see how does this all grow and develop. So barn owls have been used a lot for doing this kind of work. This is a picture of a baby barn owl, and he's wearing prism goggles that are changing his visual space. And in fact, for the lab portion of this class, there's if you guys are going and doing the labs, you'll be using some of these prism goggles, and you'll see how your overall visual switch will lead to a miscoordination of trying to hit some target, like say shooting a basket or something, if you're playing basketball. But you can adjust and change, and your brain will make some adjustments. But then when you take the goggles off, all of a sudden you're screwed up again, and you have to re-practice and retrain yourself. A lot of that is because of a misalignment that's happening within the superior colliculus. And if you wear these goggles all the time, you can change the way the map overall gets organized, especially during the course of development. And so the auditory map, we know how it normally is supposed to work, with 0 coordinated with 0, but because the ears aren't affected by the prisms, it's just the eyes, you can get this shift where the retinotopic visual map, where you would normally expect 0 degrees, is now 20 degrees, and the auditory map will shift as well in order to reflect that change so that these animals can continue to hunt like they normally should. So, and there's a ton of work on how that all occurs, which is super interesting stuff. But beyond the scope of this course, we do talk about this somewhat in my neuroplasticity class. If you were paying close attention to when I was showing this diagram of the superior colliculus sending a synapse to the PPRF, you may have noticed this other blue line going to the PPRF as well. And that's because there is another part of the brain called the frontal eye field, which is kind of an accessory portion to the motor cortex. This frontal eye field helps coordinate activity and helps coordinate the PPRF so that eyes get to the precise location. So let's talk about the frontal eye field a little bit. You can record from neurons in the frontal eye field, and they are sensitive to objects appearing within the visual field. So within the visual field, what you will see is that neurons in the frontal eye field, they will have a receptive field that they are sensitive to. So this one particular neuron that we're recording from, it is sensitive to objects here in the lower right. If you put objects here, 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 or here, you don't see any sort of activity. But if you put a target in that receptive field for that particular neuron that you're recording from, then you see activity happening within the frontal eye field that happens before the saccade actually occurs. And it appears that this, because you can do more complex experiments with monkeys where you tell the monkey to maintain its gaze at a starting point, and then when you throw up an object in the target portion, you'll start to see activity because the monkey knows he needs to make a saccade. You'll see activity increase as long as you are recording from a neuron that's within that receptive field. You'll see activity increasing there, and this is the summary of that activity here. So this is like taking all of these rasters because this is a raster diagram, and creating an average across time. And you can see that that this animal has this increase that precedes the saccade. So the cognitive understanding of, oh, there's an object in space that I'm going to need to turn my attention to, this is helping being coordinated by the frontal eye field. So if you're recording from the same neuron and you put the target in a different place within the visual field that's outside of the receptive field, you don't see any increase in activity. Tiny little bit, it appears, but it's not leading, it's definitely not proceeding with the saccade initiation. And instead, if you were to record from a different neuron that has, say, the receptive field of this new target, then you would see this kind of activity. So it's receptive field specific, and it helps coordinate the initiation of saccades. So you can see that the frontal eye field also informs this as well. Let's wrap up. Here are some of the key questions about the visual motor control system. What are the differences between saccade and smooth pursuit? Knowing some of those details about why we use saccades, why do we use smooth pursuit? What is the typical path our eyes take when scanning a scene? What are our eyes doing? When we're looking at a static scene, what are our eyes doing? What are the cranial nerves that control eye movement? What are the key neural events that regulate eye movement from the left and the right? Pay very close attention to this question because this is obviously related to diagrams. How does the fact that the eyes are mirror images of each other affect how the nervous system allows for both eyes to move in the same direction? For this question, the medial reticulatory formation is really important for that. What is the superior colliculus? What are the inputs to the superior colliculus? How does it control eye movement? What is the flow of information through the superior colliculus? What kind of information does it take in? We have sensory information coming in, motor output coming out. Do you understand how that all works? And then, last, how does activity in the frontal eye field correlate with eye movement? So this is a little bit of extra, an additional slide on the vestibular connections that help mediate horizontal eye movements. I'm not going to talk about this in any great detail. I'll leave this up so you can just kind of look at it and kind of get a sense of it. This is, again, just talking about eyes moving in horizontal space, but it involves input from the semicircular canals, because when our head is rotating, so like this is the left ear, this is the right ear, we have these semicircular canals, and you're going to have a recognition of something changing within that space. And it'll be the left. If you're moving your head to the left, you get activity from the left semicircular canal that connects to the left vestibular nucleus, which then leads to a really interesting set of connections controlling the abducens and the oculomotor nucleus, which we talked about in detail already, with the abducens cranial nerve connected to the lateral rectus, and the oculomotor cranial nerve 3, connected to the medial rectus, and then and how that coordination occurs. So I will leave it up to you to walk through the circuit if you would like, try to figure out precisely how this is actually occurring. It could be a useful exercise in understanding a little bit more about eye movement, and this is actually a really nice circuit diagram showing left and right. So in fact, maybe studying this in the context of the left and right areas for saccade control that we talked about before with the PPRF, something like this would be a really nice diagram, in fact. So yeah, I'll leave it up to you to work through, and we'll talk more later. Bye-bye.